Well, I don't want to waste anybody's time. People who got here at 2, made the time to be here at 2. I gotta say, this is the saddest attendance I've ever had at any review ever. I have 140 students and I have 6 people joining in on this Kahoot. You know, hopefully it pays off for you guys. So I'll go ahead and get started, and we are recording for people who aren't attending. I think that's what they're kind of relying on, and I hope that helps them. But yeah, just to be clear, like I know we have 6 people here, it's less likely that anybody's going to show up to the next one. I will not be giving one if nobody shows up. I'm serious. So, all right. All right. You're not going to put in your effort. I'm certainly not going to put in mine. So this is what we're going to do. We're going to go through this, their questions, but I will go over, you know, why the answer is what it is, and be sure that you guys understand. Before I move on, let me pull up the chat in case you guys start talking to me. All right. So chapter 1 first, chapter 1 and 3, just introduction to microbiology stuff. Which of the taxa is the largest? All right. The way that I would break it down, of course, is if you can remember the letters at least and be able to write them down on your scratch paper. You will have scratch paper, by the way. Then you can write those letters down and then pick out which one obviously would be the highest. So kingdom would be, well, domain is the highest, and then kingdom of what comes next. So kingdom, phylum, class, order, family, genus, species. I have to say them all like that, or else I don't remember them, and I would have to write them down. If you were to ask me this question, even right now, having said that, I have to write it down, and I'll be like, this is the one. So no worries if you need to write it down and remember. I do too. Alrighty, which of the following is not a kingdom within the domain eukaryote? Sorry, I promise I'm not trying to block everything. Okay, so when we're talking about the domain eukarya, and it's asking us about which of the following would not be, like basically just eukaryotic, our answer here would be Monera. Of course, well, we've been talking about the fungi and the protista and plantae already, and we didn't bring up Monera. So this is more or less like, can you choose this based on process of, you know, deduction here, elimination. The old classification, Monera was like bacteria and stuff. All right, moving on to chapter 3, the 5 I's. Here we have inoculation, isolation, inspection, and identification. Which of these choices is the missing one?
Inclusion.
Yep, most of you guys got this one. So it's going to be incubation. So of course, if you're going to inoculate something like we have done on our plates, the sterile plates, you have to incubate it in order to proceed with all of the rest of the other stuff. So that's the step, and they do go in order. All right, I designed a medium which only gram-positives can grow on. So what type of media is it? So what's the term for that? Okay, so yes, we would refer to that as selective medium. And just for reference sake, remember selective and differential, probably the two that you want to be the most familiar with compare and contrast on. Selective, we are selecting for a specific organism or type of organism. And then differential, it's only telling us the difference between those two. Okay. Right. So biochem stuff. Next, chemistry, we love it. Which type of chemical bond involves sharing of electrons?
Covalent. Oh, maybe actually. Here.
Yep, so that's covalent. Hopefully
Let's
these ones
go.
aren't too difficult.
Legos. Come here.
All right, we all know oil doesn't mix with water. What would oil be considered chemically, the term we would use to describe it?
Oh. Hydrophobic. Do you want to eat it on this blanket? Eat on the blanket. Good girl.
Yep, so that's going to be hydrophobic, water-bearing. It's pretty easy concept to grasp whenever you look at it that way when it's obvious like that. But yeah, that's the term for it. So water has a pH of 7. Stomach acid has a pH of 2.5. Which has the higher, excuse this misspelling, the higher concentration of hydrogen ions? Which one has more hydrogen ions?
Oh,
Okay.
dang it.
With the way that the chemistry is going to be working here. So acids, the more acid you are, the more hydrogen ions you have. And the reason the number of the actual pH number is getting smaller, remember that's basically an inverse. So anyways, yeah, so it's going to be stomach acid has more hydrogen ions. And it's going to be, if you were to look at water, equal hydrogen ions to the hydroxyl ions, they should be equal, because that's what water is. So, which is not one of the macromolecules of life? Cool, yep, vitamins. Yeah, I don't know what to tell you. It's going to be for the macromolecules of life. If you replace vitamins with nucleic acid, that would cover all 4 major groups that we talked about. What is it that gives amino acids their ability to impart specific shapes on the proteins that they comprise? Proteins are made up of amino acids. Why do they have the shapes that they do? What does it depend on? Right, good job, guys. You guys got that one. So that's the R group, the groups that are basically coming off of that chain of the amino acids, based on whether they're hydrophobic or hydrophilic and polar and charged and all of those sorts of things. That sort of stuff can affect how they interact with their neighbors and the guy across the way, and give the protein its shape overall. So that's exactly the reason why they are shaped the way they are.
Come here.
We're going to get into the bacteria. So you see irregular clusters of round cells in the microscope. What organisms might that be? Round cells.
Come here.
You can select more than one here if you want to. Right, great job. So it is going to count as Staphylococcus aureus. I meant to also have Streptococcus pyogenes. It just didn't, I don't know why it didn't work, but that does count as round. Oh, those are strips. That's why it wasn't counted. Okay, I didn't even read the whole question. I was like, round cells. Yeah, if we were going just based off of that, it could be both. But since we're irregular clusters, we can also break it down to Staphylococcus aureus. What I did want to say too is that we're going to have more questions, obviously, for chapters 4, 5, and 7, because that's the meat of what we're doing in the unit, and I hope that's all right with you guys. Talk about science. So you see chains of round cells in the microscope. Here we go. What organism or organisms might it be?
No. You want your carrot?
All right, so that is going to be Streptococcus pyogenes. Again, this is sort of the answer to what I missed on the last question myself. That's strepto being the chains that I always remember, like strips and chains. Those strip is closer to chains than staphylo. So that's how I tell the difference between the two in my mind. Whatever works for you guys.
Girl.
All right, next question. We have a multi-select, possibly more than one answer. You see chains of rod-shaped cells in a microscope. So what organisms might it be? Given this information, given the choices.
No. Sit.
Okay, so we would be able to choose here Bacillus sphaericus and Bacillus cereus. They both have bacillus in their names. That means rod. So those both could apply potentially. We would need more info though to answer, right? Okay. With the chains part. So cells that are stacked up in fence-like orientation called palisades would have to have what shape? What shape would the cells have to have in order to make that arrangement?
Good girl. Sit.
Right, so this is going to be bacilli. Just think about this question as being like, if you're making a fence, even if you don't know what palisades are, if you're making a fence-like orientation, like you're not going to be able to build the fence really that well with a cockeye, right? So yeah, the bacilli are the ones that are going to do that. I'm really not going to care too much about the arrangements of the spirals in those guys. Which appendage or appendages allow bacteria to be motile? What do they use to get around? Okay, cool. So that is going to be flagella. And remember that with the eukaryotes, which we haven't quite got to yet in the review anyways, the eukaryotes can use cilia for motion, but the bacteria do not. So just flagella for the bacteria, if they are motile. Right. What flagellar arrangement is pictured? There's going to be a few of these questions. Right, so most guys got this. It only had one flagella off of the cell, so that's going to be monotrichous. This ones are just terms we'll have to learn. We can cut some of them out as we go through it, but what flagellar arrangement is pictured? Again, okay, so the term for this one is more or less a matter of learning the term. Lophotrichous just means a tuft or a bunch at one end of the cell. That is considered a polar arrangement. So is monotrichous if you have them at one end or both ends and not just random. Those are polar, but yeah, lopho is the tuft. Right, what flagellar arrangement is pictured? I think this is the last one. Okay, so we had flagella at each end, right? So it's sort of like, yes, there are tufts involved, but it's not one tuft. And since it's at both ends, amphi means both, and that's what the correct choice would be. I don't think I have a question about peritrichus on here. I couldn't find a good image in the actual Kahoot system to put on there, but peritrichus means that they're covered randomly, completely with flagella. So that is a non-polar arrangement. That's just what that term means, in case you come across it. By what means do bacteria utilize their flagellar movement to get around? How do they do it? All right, so the majority of you guys got this. They are called runs and tumbles. No crazy words here. The runs are done by turning the flagella one way, and then the tumble is achieved by just quickly turning it back the other way. That's just how they work, so it's not a specific movement. Their flagella moves by rotation. Cool. All right, we can have more than one choice for this one. What might prompt a bacterial cell to even want to get around in its environment? What are they motivated by? Alrighty, so this chemotaxis and phototaxis. So chemotaxis, absolutely, I'm glad that most of you guys got that one. That is the biggie. Do keep in mind that when you guys take the exam, it's only going to be one choice. It's just traditional multiple choice. So there won't be situations like this. I feel like these are a little bit more challenging because you may or may not need to choose more than one. Anyways, chemotaxis can cause a bacteria want to go one way or the other. And so can phototaxis if you're a photosynthetic bacteria. So, right, which is denser and more tightly bound to the outside of a bacterial cell? Which one? All right, great job, guys. It is a capsule. I think about slime layers and I think about them being gross and all over the place anyways. A capsule, in my mind, the term anyway, would be more of a defined shape. So that kind of can help you think about it. All right, pick the cell envelope that belongs to the gram-negative bacterium. Be sure you read your choices. Move this. I don't know if it's blocking you guys. All right, so this is in fact going to be the thin peptidoglycans. That's how I would have broken that down. I would have said, okay, we have the choice between in the beginning thick or thin. So I go with the thin. Which of these two choices, lipoteichoic and teichoic acid, is there to maintain the structure of the gram-positive thick peptidoglycan layer? So that's not the gram-negative. Gram-negatives, however, on their outer membrane, because remember they have that thin peptidoglycan, but they also have that outer membrane. And on that outer membrane, they have lipopolysaccharide or LPS, which also is referred to like just when we're talking about your immune response and everything, we call it endotoxin. Endotoxin. It's the same. Right. Pick the cell envelope that belongs to the gram-positive bacterium. I did just talk about this, but I forgive you, it's Saturday. All right, so the thick peptidoglycan wall with the lipoteichoic and teichoic. I know I just said it, but that is the correct answer. We need those teichoic things to help maintain the structures, especially while we are dividing to make new cells. They do that through binary fission, just so you know. Which organism has a waxy layer containing mycolic acid on the cell envelope? Mycolic acid. Great. So a lot of you guys are also taking this exam really close to when we're doing the acid fast. And that has a pretty good reminder about which one is which and how to tell, because I talk a lot about the waxiness of it. But so yeah, that waxiness present on the outside of those cells, it makes them resistant to things. And tuberculosis and leprosy, those sorts of diseases, those bacteria are pretty resistant bacteria. And that waxiness is what gives them that quality. And it's called mycolic acid. It's that type of wax. So I know if you can remember the myco, because obviously myco and the mycolic acid matching up with one of those two choices at the bottom, the mycoplasma and the mycobacterium. And remember, the way that I remember is because plasma to me is non-shape. And so either we're dealing with mycolic acid or no cell wall. Those are your choices with the mycos, and the no cell wall would no shape. You're not holding the shape. So that has to belong to the mycoplasma. That means that the acid fast, the mycolic acid, is going to belong to the mycobacterium species, if that helps. Okay. True or false, plasmids are extra bits of RNA that give helpful abilities to bacteria. Lucky you guys that we're going to be doing this whole big experiment in the lab relating to using plasmids in order to, like, for biotech sort of stuff. And so we're going to have a whole experiment or lab about that. But that hasn't come yet. That's more of a unit 2 thing. Anyways, but plasmids are, in fact, DNA. They're just regular old DNA, like double-stranded DNA. They're just smaller circles of it than the normal bacteria genome. And bacteria do not require it to live. So, but they might get, nice traits from it. They're just little extra circular bits of DNA and RNA. Okay, endospores. I love them. What's the term for they're becoming when they're becoming metabolically active again? Man, I really just typed these up, didn't I? All right. The process. All right, so the process of going from the endospore, that protective structure, back into the metabolically active cells, that's called germination, that process is. I think there's another question relating to the endospores next, so I don't want to give away too much just yet. I hope there is, because I have more to say on endospores to remind you guys. Yeah, here it is. What's the term for the metabolically active bacterial cell? You know that the endospore, we just talked about that, at least you should have been paying attention, is the dormant one. So it's not going to be the green. Okay, so the term for these guys, the metabolically active bacterial cells just by themselves, those are vegetative cells. So remember, we're going to be having vegetative cells, happy living, dividing. Things go south, they need nutrients, so they want to become protected and wait for things to get better. The process of going through that is sporulation. And so we're forming endospores there. We form the endospores, we're an endospore, We are dormant. We are not metabolically active. We're just waiting for that trigger to be induced to come out of our little shell, our cortex. And when we go from being the endospore back into the vegetative cell, that's called germination. Yeah, that's all I have to say about it, right? Yes, I think so. I think that's it. Yeah, if you guys have any questions, of course, please let me know. Okay, why should we give a flying hoot over endospores? What's the deal? Why do we care? It might be more than one decision, more than one choice, by the way. All right, so this one was all of them. Just trying to get the point across that it's not just because like they're hardy hanging out in the soil make you sick, but they can actually affect our lives in a lot of different ways. So make it difficult. All right, be aware of them. So true or false, archaea are extremophiles. Yeah, so they are. For our purpose in this course, just you can associate it directly with being extremophiles. So you guys are doing it. Okay, you're sick with Campylobacter. It is fastidious, gram-negative, spiral-shaped, and microaerophilic. So what term would it apply to it? Okay, so this question, obviously, you have to be able to know that all of these terms are referring to the structure of like the cell envelope of the organism. And if you aren't aware of that, you won't be able to pick out what's important in the question. So it's given us all this information, most of which we don't need. And then we look at our choices of the terms to choose from. And like to me, okay, firmacutes, right? Firmacutes, firm, that's the gram positives. Remember Mendoza cutes? Joey Mendoza was cute. That is a long time ago. That's archaea. Then we have the gracillocutes, those are the gram negatives. That's the next G gram. And then the tenericutes are what's left. That's the ones without the cell wall. So the mycoplasma, for example, would fall into that. So Campylobacter, it's gram negative, it would fall into gracillocutes. So we really only needed the gram negative part of this, but you do have to know what those terms are referring to in order to pick that information out. There are a few questions like this on the exam that you'll have to be aware of what you're choosing from, and be aware of, understand your surroundings in order to answer the question. So be sure you do take time to understand what you're dealing with. So when trying to identify an unknown bacteria, what's the best to start with? What do you think would be the best one to start with? It's unknown. Where should you begin? And yeah, no, it's not a trick question. Good job, guys. So yeah, and I never specifically said this, but clearly like that's where we started in our lab, and we're doing an unknown project. But in real life, yeah, you would want to divide it into gram positive or negative first. If it were gram negative, let's say, like the Campylobacter we just talked about, then you would not waste your time doing an acid-fast stain or an endospore stain. Both of those only apply to gram-positive bacteria. Acid-fast, remember, that's just going to be mycobacterium. All mycobacterium are gram-positive. That isn't a difficult one. An endospore is bacillus and clostridium. I just happen to know that those are both gram-positive. So yeah, so you wouldn't even need to do those. If you're a microbiologist, you would know that. Saves you time. So gram stain, exactly. Gram stain is where you start. It's your bread and butter. Okay, that was the bacteria. It seems like some of this stuff was just yesterday, but Let's get into the eukaryotes again. True or false, all eukaryotes have mitochondria. Mitochondria. Mitochondria is what it's supposed to say. So this is true, and we can kind of infer this just by knowing that eukaryotic cells make their energy using mitochondria. And I didn't talk about any other way that they can make their energy. They make their energy in mitochondria. So yeah, so all of them have to have that. Just kind of infer from that, but yes, is correct. Okay, true or false, eukaryotic cells multiply to make copies of themselves via meiosis. Is that true or false? Making copies of the original cell, is that meiosis? All right, so the answer for this one is false. The term for making copies of the original cell to make identical daughter cells, that's mitosis. That's going from, 5 chromosomes, and your daughter cells each have 5 chromosomes, or something like that. Maintaining that. If you are making gametes for sexual reproduction, the process for going to do that, that's meiosis. So regular copies, mitosis. I know they have to sound the same, right? Many fungi are saprobes. What are saprobes? Jeez, man. All right, so yes, so saprobes is just a term you would have to learn and study. This is definitely a flashcard moment to learn this one, but obtaining nutrients from decomposing dead matter. That's what saprobes is. They break down the dead things. Decomposers. What is the woven, intertwining body or colony of mold called? You know, it's like I didn't even try to spell. The whole body of that mold. Okay, so a sporangium, just so that we're clear on this. The sporangium in the red choice, that is a term relating to bacteria. And it's the endospore forming bacteria while the vegetative cell is generating an endospore within it. That outside portion is called the sporangium. So it doesn't have anything to do with our mold. Okay. So we would be able to pick that out. Anyways, the hyphae, the yellow one, that's just like each individual like thread-like growth off of them. So, if you had mold and it was hairy or fuzzy looking or whatever, each one is a hyphae and they can branch. If they're segmented, the segments are separated by septae, which is the green choice. Mycelium refers to the major like shape of it. So that whole ball of mold growing on your piece of cheese or whatever, that whole thing is called the mycelium. Good. True or false, fungal spores are very similar to bacterial endospores. Wow, we're really divided, house divided here. So they are not. So fungal spores are made for reproduction purposes only. And remember that bacterial endospores are dormant. They are not reproducing at all. So they're just waiting. So the fungal spores, nice, happy, good, everything's great. And now we're reproducing type of situation. Okay. Releasing happy spores into the environment and they're going to go grow mold on your other piece of bread. What type of fungal reproductive body is not enclosed by a spore-bearing sac? So thinking about which one has the covering here. All right, so I have just talked about just a little bit ago, a couple questions ago, sporangium being related to bacteria, right? So we should be able to kick out blue and green from that alone. So then our two choices should be red and yellow, conidiophore versus sporangiophore, right? And I know that we had talked about conidiospores and sporangiospores, but each of those spores belong in a phore as the whole structure. So is that structure covered or uncovered? The covered one is choice yellow, that's the sporangiophore. And the uncovered one, where we're just like flowering out all the time, spores flying out into the wild blue yonder, and they don't have to bust out, that's the conidiophore. The conidiophore is uncovered. All right, you may choose more than one, which belong to the kingdom Protista. Which of these belong to Protista? You'd think that I hadn't actually looked over this before I did this without any mistakes, but I actually did. I was just looking over the content and not checking the spelling. I apologize. All right. Yes, it's going to be algae and protozoa. Anything that doesn't have true tissues will fall into that. Protista happens to be the algae and the protozoa. We have algae to thank for most of our, oxygen in our environment. They do this as plankton primarily. Keep in mind that fungi, they have their own kingdom. That's their own kingdom. All right, which of the following is not a means by which protozoa move around? Let's see. I can hear my dogs barking. Who knows why? All right, so that valvular translocomotion, that's going to be the choice. The protozoa, or don't use that. I don't know. I think I may have just like tried to make up a word that sounded sciencey or something, or like Googled something that didn't have to do with protozoa. I don't know where that word came from. It's not applying at all to what we learned. We did learn about cilia, the little oar-like motions that they beat the water with. They use it to feed, and they can use it to move around. We did learn about flagella in eukaryotes. Remember, they can move with whip-like, and they're made with the microtubules inside and coated with the membrane, part of the cell. Just continuous covering. And then we have these pseudopods. Those are the fake feet, pseudo, fake pods. That's supposed to be like a podiatrist feet. So that's like what we have with amoebas. So protozoa, amoebas are protozoa is what I'm trying to say. Nice. All right, which is the term for the active, free-living form of protozoa? Why are they barking? All right, so the free-living form of the protozoa is called the trophozoite. When we think of the bacteria, we have the vegetative cell and the endospore, right? And I know that was like what we learned first, so we can equate the protozoa version back to that. So the vegetative version, that happens to be the trophozoite, and the endospore version would be the cyst. And so how would you remember, if you can remember that's trophozoite or cyst, how would you remember which is which? Well, remember that troph means to eat. And if you are eating, you are not dormant. You can't eat when you are dormant. So it would have to be the trophozoite eating. That's one that is free living and metabolically active. The cyst is the other one. We know endospore and vegetative are terms for the bacteria. Cool. That clears that up. You may choose more than one here. What is parasitology a study of? It's kind of a tough one if you're just trying to figure it out by the word itself and no other information.
Record.
All right, that's okay. This is not a term that I expect you guys to necessarily know for the test itself. I did mention it in maybe a class or two, and it's not something I would expect you to know if it's not on the sheet. But moving forward, especially as we learn about the diseases, parasitology. I don't know why, because parasite is a term that means that the pathogen, let's say, whatever it is eating and getting energy from a host that is being harmed by that organism. And that organism that's doing that, we call that a parasite, just like a tick or a leech or something. And on the microbe scale, which is what we're talking about here, you could think you could apply that to pretty much anything. But the actual field of parasitology deals with helminths and protozoa. So there you go. So again, this is more something that's going to be important going forward. I'm not going to put that on the test. I just needed you guys to kind of be familiar with the term. So Ascaris lumbricoides, it causes the most helminth infections and is known as a large intestinal roundworm. And what would it fall into? Using this information I just gave you. Okay, these are the major classifications of helminths based on essentially their body structure, whether they are flatworms or roundworms, and if they are flatworms, whether they are a tapeworm or a fluke. So these are our choices here. That's what these words at the bottom actually are referring to. So because we obviously definitely know that, okay, so now we just want to know what the shape of this organism is. So go back up to our question. It tells us that it is a roundworm. So a roundworm, the term for roundworms, and that part, then you have to know that nematoda is the roundworms. Platyhelminthes is the flatworms. And then we have cestoda as the tapeworms and trematoda as the flukes. So those are just, once you get down to that point, flashcards. That stuff it is for flashcards. Okay, that was bacteria. That was eukaryotes. We're done with our cell-based microbes. We're going to move on to our acellular microbes. So that's chapter 7. We just went over this. Viruses. They are obligate intracellular parasites. I'm sure you guys remember that. So what is a capsid in a virus? What's the capsid? Okay, so all viruses have to have two things at a bare minimum. That's going to be your capsid and your nucleic acid, right? And I could go on all day, you know this, about what all of that could include. But as long as you have that capsid protein coating and the nucleic acid, that's your base. Together they form the nucleocapsid. So yeah, the capsid is that protein covering over the genome. All right, true or false, every virus has an envelope. Good job, guys. Yes, this is going to be false. So there are plenty of viruses that don't have the envelope, the membrane covering. They can just be a capsid in a genome. And think about the viruses that infect the bacteria, we'll just call them that for now because there's a question. They do not have an envelope on them, right? So what is the purpose of the viral spikes? What are they doing for the virus? Okay, so yeah, so that helps them dock and enter the host cells, right? It allows them for host selection, host range really is the better term if I'm being sciencey. But yes, so it determines your host range, what cells you can enter into and what organism types and species and whatever. That's when I was talking about describing to you guys, that's our key. The spikes are the key and the lock is the protein on the cell. They interact via this docking, spike docking. What is a virus like poliovirus called if it doesn't need an envelope? Right, naked. I mean, that's just the term you'll have to know. Capsamere, that's a protein, just like a protein unit. They come together to form the capsid. They're just like repeating units to make that up. And then we have the icosahedral is the shape that a capsid can have, one of the shapes it can have, that or helical. Yeah, lonely is just, I don't know, all of us at some point, right? Okay, this is a quiz question, a multi-select quiz question. Which of the following are types of genome viruses could have? Yeah. What types of genomes can they have? Multi-select. Yeah, once you guys get in this one. So they can have any double-stranded or single-stranded of RNA or of DNA. That's basically it. And remember, we talked about positive and negative strand of negative, sort of positive and negative sense as well. So what is the term for when viruses bind onto their target cells to initiate infection? What was that term? And the steps of viral infection. So the term for this is adsorption. So when they're trying to bind to initiate, that step is called adsorption. For the budding, remember, this is whenever the viruses are going to be building and structuring themselves in the middle of the infection process, getting ready to release. So they're pushing out and wrapping themselves with the envelope and pinching it off behind them. That's budding. Where does synthesis of viral components occur for viruses with DNA genomes? Not a trick question. Okay, most of us should pick endoplasmic reticulum, and that's okay. I understand where this is coming from. So DNA multiplication in general is going to be happening in the nucleus. It has to replicate the DNA there. It even has to make a messenger RNA in the nucleus. The messenger RNA, just like our messenger RNA, then goes to the endoplasmic reticulum to get translated into protein by the ribosomes, yeah? But that protein has to come right back into the nucleus. So the only time we're outside of that nucleus is when we're making the protein part. So most of it is going on in the nucleus, and man, if they could just do all of it in the nucleus, they probably would. So yeah, nucleus, just remember that if you see DNA viruses, that they're going to be doing the nucleus, the RNA ones, cytoplasm. Okay, cytoplasm, that's where the endoplasmic reticulum is. Where does synthesis of viral components occur for viruses with RNA genomes? Okay, so our answer here is cytoplasm. And the reason, again, that it is not endoplasmic reticulum, not all of the viral components are being made in the endoplasmic reticulum, if any, because you can make protein without the endoplasmic reticulum. You can have ribosomes that are not on the endoplasmic reticulum. Either way, the endoplasmic reticulum is still also in the cytoplasm. So technically all of it is going to be going on in the cytoplasm, including within the ER, but not all of it's happening in the ER, right? So cytoplasm. Just remember that. Nucleus, cytoplasm. And that's how we can basically divide a eukaryotic cell, the nucleus, because that's really the important structure of it, right? And then around, outside of it, all of that is the cytoplasm. And there are organelles within it, like the ER. That helps. Okay, how do eukaryotic envelope viruses get out of their host cells? Your regular old COVID, how are you going to leave the cell? Yeah, once you guys got this one, it's going to be budding, that pinching off with the membrane surrounding it, or exocytosis, which is literally the cell using vesicles to empty the contents of the vesicle outside of the cell, like it would normally if it was making stuff that it wanted outside of the cell, a toxin or a signaling molecule. So how do bacteriophages get out of bacterial cells? Okay, so bacteriophages, the viruses that infect bacteria. They'll come around like they're little alien spaceship cells, and they'll land on the bacterial cell and squat down and inject their genome straight into the bacterial cell. They don't enter the bacterial cell, right? So they land, inject their genome in, and then the bacterial cell starts making the components, starts building up all new bacteriophages. And it just keeps doing that until it busts open. So there's no budding because they don't have a membrane, because they're so complicated with those legs and everything. Look at the spaceship. You won't cover that up. The whole point is that it has to be able to function. So you can't cover that up with a membrane. But yeah, so they just bust out. Always. Bacteriophages. In eukaryotic viral infections, what is the term for viral DNA that has been incorporated into the host cell genome DNA? You have HIV and DNA from HIV that's incorporated into your genome. Now, what do we call it? Alrighty, so this one is provirus. I think the next question is going to talk about the prophage. I'm not entirely sure, but I'll just go ahead and mention it now, whatever, it'll be a free point if it is. But if you hear provirus, that's going to be talking about eukaryotic viruses. Prophage, always bacterial viruses, and they have their own terms related in that way. That makes sense. So they have their own thing going on. Provirus, prophage. Right, here we go. In bacterial phage infections, what's the term for phage DNA that has been incorporated into the host cell genome DNA? We can think about it as, provirus and prophage, they're the same thing. It's just one is eukaryotes and one's bacteria. We know the bacterial ones are called phage, so prophage. What is the term for the type of phage that is capable of switching between lysogenic and lytic? What's the term for that? This is a question that is pretty similar to the kind that I will have on the test. And when I say that is because these are terms that kind of all apply to viruses. You need to know which one is which. All right, great job, guys. This is in fact the temperate phage. The temperate phages can switch between the lysogenic and the lytic phases. Lytic, making virus normal, popping them out of the cell. Lysogenic, inserting the DNA into the genome of the bacterium as now it's a prophage, and then waiting for induction to switch back to lytic, right? Start making viruses again. Induction is just anything that causes that to happen. So yeah. And remember, when we have our DNA inserted in there as the prophage during the lysogenic cycle, it can have genes with it that help the bacteria be more pathogenic. I know that these questions are coming up after this, it has to. All right, when a eukaryotic virus hides out and lies in wait to resurface at a later date, that is known as what kind of infection? Okay, so yeah, the correct term for this is going to be latent. Remember, lysogenic is kind of like latent for bacteria. Lytic, that's for the bacteria viruses that are going to be just replicating. So for eukaryotes, when we're waiting, hiding in wait, that's going to be called latent. That's their term for it. Similar things going on. The lytic phase is the phase where the phage is synthesizing new phage virions. Is that true or false? We're making new virions, they're infectious. Is that called the lytic phase? All right, so that's true. Yeah, I've been talking about the past couple of questions. So yes, it's just the lytic phase, new viruses being made and bursting out, right? Lysogenic, insert the prophage into the DNA and wait for induction. True or false? Lysogenic conversion occurs when a prophage gives bacteria pathogenic traits. Is that the term for? I was just talking about it. Prophage gets inside of the DNA of the bacteria, gives you new traits, is the term lysogenic conversion. So yeah, this one's true. Definitely lysogenic, we know that. And we are converting that bacteria from whatever it was before to now being more pathogenic. Think of it that way. When a virus causes cancer to form in a host, this process is known as, what is the term? I know you guys probably see this and probably think, oh, oncovirus. Yes, it would be an oncovirus. But what is happening to the cells? Okay, so the correct term, whenever an oncovirus infects a cell and causes cancer to happen, that process is called transformation. Okay, transforming. I know it seems like a basic word, but that is really what it's called. All right, scientists can choose more than one for this. Scientists culture viruses in tissue culture to study them and create vaccines. How do they know that the virus is growing? What are some ways they can find out that there's actually virus in there? And a lot of time for these, especially, I'm talking about looking at it straight on. So the answers are going to be blue and yellow. So visible cytopathic effects and formation of plaques. If we're talking about eukaryotic cells, we're mostly talking about the blue, the cytopathic effects. The cells will detach from like the plastic bottle. They'll round up. If you do look at them under a microscope, they'll be like rounding up. They won't be adhering to one another anymore, and that sort of stuff. So behaving strangely after the viral infection, whereas before they were infected, they were cells doing cell things. All right, that's cytopathic effects, anything that causes the cells to behave irregularly from what they should be. So the formation of plaques is primarily talking about bacteria, not entirely, but that's what I always think of anyways, that lawn of bacteria, putting the viruses on there, diluted, you think there's virus in there, dilute it, put it on your lawn, and put it to grow overnight. And if there's empty circles, those are the plaques where like one virus became many viruses, became many viruses, became many viruses outward. Those are plaques. Okay, so we're looking for blue and yellow. Tumor formation in the culture isn't really a thing. These are kind of already immortalized cells, so they're not going to form tumors, and they're definitely not going to. If they started growing up on top of each other, I would think that would be a cytopathic effect. Anyways, and then we have the visible viral particles. You aren't going to be able to see viruses in a typical microscope or while you're doing cell culture. Most of these other ones, you can actually see the effects. True or false, interferon is a natural cytokine that allows cells to signal one another to prepare to fight incoming viral infections. True or false? Right, so this is that molecule I was talking about. Your neighbor lets you know that there's, a burglar coming, a virus coming, and to get ready. This is interferon. You're going to interfere with the viral infection cycle. All right, I think this is the last question, last actual question. Which of the following are actual spongiform encephalopathic diseases caused by prions? Some of these we didn't talk about, so just to be fair. Oh yeah, I'll just go ahead and tell you guys that it's all of them. Okay. All of them are. Because I know I didn't talk about fatal familial insomnia. That's my bad. So yeah. Scrapie, sheep version of mad cow. Chronic wasting disease, deer version. Kuru is the cannibal human version. The regular human version, Creutzfeldt-Jakob. If we got Creutzfeldt-Jakob by eating beef or something similar, variant Creutzfeldt-Jakob would occur. So VCJD. And then fatal familial insomnia is a hereditary insomnia disease. You die from not sleeping, literally. So that's kind of crazy. All right. So that's all I have for the Kahoot for today. I hope that was helpful for some of you to go through and refresh your mind on this information. While you're studying, if you guys have any questions, please, reach out to me and I'll try to help you with what I can. If you guys have any questions now, of course, I'll be happy to answer what I can as well. So other than that, I'll probably hang out here for a little bit and wait for that. And like maybe just a couple minutes. And as you guys can kick off, I will just, probably kick off myself. So I'll see you guys on test day. Yeah, I'm happy to do this. It's fun for me too.
Can you hear me?
Yes, I can.
I had a question. It's not over the review, but I was trying to upload my chapter 7 from the lab, and it won't let me upload it in the top hat. How do you want me to submit that? I have access to it, but it's just grayed out. Like it won't let me to upload it.
Which class are you in?
I am in. Let me go back.
Tuesday. It's okay. It's Tuesday morning or
Tuesday, Thursday evening,
Okay, at 4 p.m. lab or the late lab?
the late lab.
Okay, so I think, when's the last time you tried it? I ask, because I like right before this, I updated the due dates, but I don't know if you.
Oh, okay. I had tried it this morning. I'll try it again now.
Yeah, let me know if it doesn't work though. So I think my Tuesday, Thursday, 4 p.m. people, I didn't do it the right way, but if you didn't upload it, you should be able to anyways. But yeah, it should be able to work now. I literally just changed it. This was me being, I don't get up before noon, so. And I didn't have my computer with me until today. So, and I can't do it on my phone. I don't know why that I can't get on Top Hat on my phone. I'm the teacher. You'd think I would need to, but I don't have access on my phone. So I apologize for that to you guys, but hopefully it works now. I was told it would.
Okay, yeah, I figured it out. It's gonna let me upload it.
Okay, yeah, like I said, I just changed it. So yeah.
Okay, thank you.
Yeah, no problem. All right, everyone, I'm gonna go ahead and go. Like I said, please email me if you do think of anything. I'll be happy to help, honestly. So have a good one.